The goals of the Clarity Challenge describe the problems people, including us, are facing when applying NGS sequencing data to clinical diagnostics, so of course we wanted to take part. Before applying, however, we had to set up a team with expertise in many areas including IT, computational biology, human genetics, and pediatrics. The first team member we called was Saskia Biskup, human geneticist and CEO of CeGaT. She was excited right away and expanded our team by the pediatrician Peter Freisinger and the pathologist Sven Perner. Once the challenge started, Children's provided medical information and sequencing data for each of the 3 families. When analyzing these huge data sets, we decided to keep things as simple and transparent as possible. The main problem thereby is that each human being has millions of variants, and we have to filter out all but the one that's disease-causing. So the first filter removes all variants that don't affect the protein sequence. And even though this removes about 99%, we are still left with thousands of variants. So next we remove those that occur frequently in the population. Then the so-called pedigree filter removes all variants that are not in line with the expected inheritance pattern. And the last filter removes all variants that cannot be linked to the given disease. This is based on CeGaT's expert-created disease panels and literature mining from Genomatix. In several cases, this approach directly pinpointed one presumably causal variant out of the millions to start from. When a potential causal variant is found, the medical report is generated by providing the filter settings and the causal variant itself. To enable medical staff applying this approach in an access-controlled setting, we've developed a tool called GeneGrid to allow for using all filters and overlay the medical information.
When I was asked to participate in the Clarity Challenge, I immediately said yes. As a human geneticist closely collaborating with clinicians, the potential and the impact of next-generation sequencing in a daily clinical setting is absolutely clear to me. Every day we solve a case that has not been solved for many years because the technology was not available is a good day. Every gene that we discover as the cause of the disease is a potential future therapeutic target. Next-generation sequencing is not the future. It's today. Have a look at the results of the Clarity Challenge.